Hello friends, today we will talk about enzyme specificity, role of binding energy in enzyme specificity and interactions between enzyme and substrate. And the main objectives of today's lecture are to understand the concept of enzymes, to study enzyme specificity, to study role of binding energy in enzyme specificity and catalysis, and to study the enzyme-substrate interaction. First of all, we will study about enzymes. Enzymes are proteins that increase the rate of reaction by lowering the energy of activation. They catalyze nearly all the chemical reactions taking place in the cells of the body. In the absence of an enzyme, the reaction may hardly proceed at all, whereas in its presence, the rate can be increased up to 107-fold. Enzyme catalyzed reactions usually take place under relatively mild conditions as compared to the corresponding chemical reactions. Nearly all enzymes are proteins, although a few catalytically active RNA molecules have been identified. The active site of an enzyme is the region that binds the substrate and converts it into the product. It is usually a relatively small part of the whole enzyme molecule and is a three-dimensional entity formed by amino acid residues that can lie far apart in the linear polypeptide chain. The active site is often a cleft or crevice on the surface of the enzyme that forms a predominantly non-polar environment, which enhances the binding of the substrate. The substrate is bound in the active site by multiple weak forces such as electrostatic interactions, hydrogen bonds, van der Waals bonds, and hydrophobic interactions, and in some cases by reversible covalent bonds. Having bound the substrate molecule and formed an enzyme-substrate complex, catalytically active residues within the active site of an enzyme act on the substrate molecule to transform it first into the transition state complex and then into the product, which is released into the solution. The enzyme is now free to bind another molecule of substrate and begin its catalytic cycle again. Now the enzyme specificity. Enzymes are highly specific, catalyzing the reaction of a specific substrate or a specific group of substrates. In addition, enzyme activity can be regulated, varying in response to the concentration of substrates or other molecules. If one of the enzymes needed by a cell is absent or non-functional, then one of its chemical reactions can't proceed. No other enzyme can substitute for the missing one. Enzymes having varying degrees of specificity for substrates. They may recognize and catalyze a single substrate, a group of similar substrates, a particular type of bond. In general, specificity can be categorized as first, group specificity. In this type, a general group of compounds may serve as substrate. The enzyme will act only on molecules that have specific functional groups such as amino, phosphate, and methyl groups. For example, carboxypeptidases, pepsin, aminopeptidases, and hexokinases. And second is absolute group specificity. In this type, the enzyme will attack only one single substrate. For example, trypsin hydrolysis peptide bonds, but will attack peptide bond. And third is relative group specificity. In this type, the enzyme will act on certain chemical grouping in several substrates. For example, glycosidases on glycosides, alcohol dehydrogenases on primary alcohols. And the fourth one is stereochemical specificity, which includes specificity for optical isomers. For example, certain amino acid oxidases will act only on amino acids of the L series in preference to those of the D series. Specificity for geometrical or cis-trans isomers. For example, fumarase will add water across the double bond of the trans isomer fumaric acid. but is completely inactive in regard to the cis isomer malic acid. Enzymes of the Krebs cycle in general exhibit cis-trans specificity. Fifth one is linkage specificity. The enzyme will act on a particular type of chemical bond regardless of the rest of the molecular structure. For example, chymotrypsin catalyzes the hydrolysis of peptide bonds, alcohol dehydrogenase, malate dehydrogenase, and the lactate dehydrogenase reactions. Now we will see what is substrate specificity. The properties and spatial arrangement of amino acid residues from the active site of an enzyme will determine which molecule can bind with the substrate for that enzyme. Substrate specificity is often determined by chains in relatively few amino acids in the active site. This is seen in the three digestive enzymes like trypsin, chymotrypsin, and elastase. These three enzymes belong to a family of enzymes called the serine proteases. Serine, because they have a serine residue in the active site that is critically involved in catalysis, and protease because they catalyze the hydrolysis of peptide bonds in proteins. The three enzymes cleave peptide bonds in protein substrates on the carboxyl side of certain amino acid residues. Trypsin cleaves on the carboxyl side of positively charged lysine or arginine residues. Chymotrypsin cleaves on the carboxyl side of the bulky, aromatic, and hydrophobic amino acid residues. Elastase cleaves on the carboxyl side of residues with small uncharged side chains. Their different specificities are determined by the nature of the amino acid groups in their substrate binding sites, which are complementary to the substrates that act upon. Thus, trypsin has a negatively charged aspartate residues in its substrate binding site, which interacts with a positive charge on the lysine and arginine side chains of the substrate. Chymotrypsin has amino acid residues with small side chains such as glycine and serine in its substrate binding site that allow access of the bulky side chain of the substrate. In contrast, elastase has the relatively large uncharged amino acid side chains of valine and threonine protruding into its substrate binding site, preventing access of all but the small side chains on glycine and alanine. Now, the principles of catalytic power and specificity of enzymes. Enzymes are extraordinary catalysts. The rate enhancement they bring about are in the range of 5 to 17 orders of magnitude. Enzymes are also very specific, readily discriminating between substrates with quite similar structures. Covalent interactions between enzymes and the substrates lower the activation energy and thereby accelerating the reaction by providing an alternate lower energy reaction path. Much of the energy required to lower activation energies is derived from weak non-covalent interactions between substrate and enzyme. What really sets enzyme apart from most other catalysts is the formation of a specific enzyme-substrate complex. The interaction between substrate and enzyme in this complex is mediated by the same forces that stabilize protein structure, including hydrogen bonds and hydrophobic and ionic interactions. Formation of each weak interaction in the enzyme-substrate complex is accompanied by the release of a small amount of free energy that stabilizes the interaction. Actually, two fundamental and interrelated principles provide a general explanation for how enzymes use non-covalent binding energy. First, much of the catalytic power of enzymes is ultimately derived from the free energy released in forming many weak bonds and interactions between an enzyme and its substrate. This binding energy contributes to specificity as well as its catalysis. And the second is weak interactions are optimized in the reaction transition state. Enzyme active sites are complementary not to the substrates but to the transition states through which substrates pass into. They are converted to products during an enzymatic reaction. Now, the role of binding energy in enzyme specificity and catalysis. The energy derived from enzyme-substrate interaction is called binding energy. Its significance extends beyond a simple stabilization of the enzyme-substrate interaction. Binding energy is a major source of free energy used by enzymes to lower the activation energies of reactions. Binding energy accounts for the huge rate accelerations brought about by enzymes. The energy available for formation of a single weak interaction is generally estimated to be 4 to 30 kilojoules per mole. The overall energy available from a number of such interactions is therefore sufficient to lower activation energies by the 60 to 100 kilojoules per mole required to explain the large rate enhancement observed for many enzymes. The same binding energy that provides energy for catalysis also gives an enzyme its specificity. The ability to discriminate between a substrate and a competing molecule specificity is easy to distinguish from catalysis. But this distinction is much more difficult to make experimentally because catalysis and specificity arise from the same phenomenon. If an enzyme active site has a functional group arranged optimally to form a variety of weak interactions with a particular substrate in the transitional state, the enzyme will not be able to interact to the same degree with any other molecule. For example, if the substrate has a hydroxyl group that forms a hydrogen bond with a specific glutamate residue on the enzyme, any molecule lacking a hydroxyl group at that particular position will be a poor substrate for the enzyme. In addition, any molecule with an extra functional group for which the enzyme has no pocket or binding site is likely to be excluded from the enzyme. In general, specificity is derived from the formation of many weak interactions between the enzyme and its specific substrate molecule. The importance of binding energy to catalysis can be readily demonstrated. For example, the glycolytic enzyme triose-phosphate isomerase catalyzes the interconversion of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Binding energy can be used to overcome all these barriers. First, a large restriction in the relative motions of two substrates that are to react or entropy reduction is one obvious benefit of binding them to an enzyme. Binding energy holds the substrate in the proper orientation to react as substantial contribution to catalysis. Because productive collisions between molecules in solution can be exceedingly rare, substrates can be precisely aligned on the enzyme with many weak interactions between each substrate and strategically located groups on the enzyme clamping the substrate molecule into the proper positions. Second, formation of weak bonds between substrate and enzyme results in resolution of the substrate. Enzyme-substrate interactions replace most or all of the hydrogen bonds between the substrate and water. Third, binding energy involving weak interactions formed only in the reaction transition state helps to compensate thermodynamically for any distortion, primarily electron redistribution, that the substrate must undergo to react. Finally, the enzyme itself usually undergoes a change in conformation when the substrate binds, induced by multiple weak interactions with the substrate. The motions can affect a small part of the enzyme near the active site or can involve change in the positioning of entire domains. Typically, a network of coupled motions occurs throughout the enzyme that ultimately brings about the required change in the active site. Induced fit serves to bring specific functional groups on the enzyme into the proper position to catalyze the reaction. The conformational change also permits formation of additional weak bonding interactions in the transition state. In either case, the new enzyme conformation has enhanced catalytic properties and interactions between enzyme and substrate. Enzymes were structurally complementary to their substrates so that they fit together like a lock and a key that was proposed by Emil Fischer in 1894. specific interaction between two biological molecules is mediated by molecular surfaces with complementary shapes that has greatly influenced the development of biochemistry, and such interactions lie at the heart of many biochemical processes. The lock and key hypothesis can be misleading when applied to enzymatic catalysis. An enzyme completely complementary to its substrate would be a very poor enzyme. Some weak interactions are formed in the enzyme-substrate complex, but the full complement of such interactions between substrate and enzyme is formed only when the substrate reaches the transition state. The free energy, that is the binding energy, released by the formation of these interactions partially offsets the energy required to reach the top of the energy hill. The summation of the unfavorable positive activation energy delta G and the favorable negative binding energy delta G results in a lower net activation energy. Even on the enzyme, the transition state is not a stable species but a brief point in time that the substrate spends atop an energy hill. The enzyme catalyzed reactions is much faster than the uncatalyzed processes because the hill is much smaller. Weak binding interactions between the enzyme and the substrate provide a substantial driving force for enzymatic catalysis. The groups on the substrate that are involved in these weak interactions can be at some distance from the bonds that are broken or changed. The weak interactions formed only in the transition state are those that make the primary contribution to catalysis. The requirement for multiple weak interactions to drive catalysis is one reason why enzymes and some coenzymes are so large. An enzyme must provide functional group for ionic hydrogen bond and other interactions, and also must precisely position these groups so that binding energy is optimized in the transition state. Adequate binding is accomplished most readily by positioning a substrate in a cavity, that is the active site, where it is effectively removed from water. The size of proteins reflects the need for superstructure to keep interacting groups properly positioned and to keep the cavity from collapsing. This brings us to the end of lecture. Hope you have enjoyed and understood. Thank you and goodbye.